So this session grew out of a session we had last year. It wasn't quite the same subject, but the idea was there was this problem with research antibodies. And so as a result of that, myself and Andreas Pluchthung and over 100 other people wrote a commentary in Nature, which I'll mention briefly. And so there's clearly a problem with research antibodies. For those of you that were in the session this morning, you heard Joe Gray talk repeatedly about the problems of getting reasonable antibodies. So what I'm going to address is provide a bit of an overview. I'll touch on some of the work that some of the others in this session will be speaking about. And I must say that my opinion on this has matured, I guess some people might say, taking commercial considerations taking commercial aspects into consideration as well. So let's start with a scary story. This is clinical trials with a target called ERCC1. First, okay, so it was shown some time ago that low levels of this protein detected using this antibody seemed to predict potential benefit from cisplatin-based adjuvant therapy in non-small scale lung cancer. And then something happened. A new lot of 8F1 produced by the same manufacturer came out, and tumors that were previously described as low ERCC1 now showed high staining, and now there was a slight benefit to high staining as opposed to low staining before. Then it turned out that this antibody 8F1, in addition to recognizing ERCC1, it also recognized this other antigen called CCT alpha. And in some tumors, but not all, it was a recognition of this second target, CCTF alpha, that was responsible for the major immunoreactivity of this 8F1 antibody. And furthermore, high levels of this target and not ERCC1 predicted longer disease-free and overall survival in some lung cancers. Now, the recognition specificity of this antibody was tied between these two different targets, and it was really rather difficult to sort out exactly what was going on. There's two important points here. First, this is a monoclonal, and it's recognizing two targets. Other people subsequently found that the recognition of two targets by this monoclonal was not due to two chains, which I'll mention, but it was actually an intrinsic property of the antibody. And the second problem is that the property of the antibody has changed with a new lot. And this is a monoclonal. Monoclonals are supposed to be really, reproducible and don't change. And Glenn Begley, who published a paper a few years ago where he tried to repeat 53 so-called landmark preclinical studies in cancer, was only able to repeat 6 of these. 47 failed. And I've spoken to him subsequently. He said that bad antibodies and siRNA are the two biggest culprits and one of the reasons why so many of those studies failed. So people have talked about this. So David Rim, who'll talk later, wrote a very good review in Biotechniques on antibody publication. I've highlighted here, furthermore, I can't read it here. Furthermore, for commercially available antibodies, it's clear that what's on the label doesn't correspond to what's in the tube. And down here, this was the Journal of Comparative Neurology, and the editor wrote, "Blind faith that the antibody will stain whatever the manufacturer claims is not consistent with good science." And so the problems are that antibodies don't recognize their correct targets, and perhaps more often they do recognize incorrect targets. And it's this dichotomy between these two problems that leads to many of the problems associated with research antibodies. Can it be quantified? And so Matthias Ullen in 2008 looked at a bunch of different antibody manufacturers going from A here to AD here. He didn't reveal the names of these individual manufacturers. I think that was a condition under which he managed to get the antibodies. And you'll notice that some companies, company A here, produces excellent monoclonals, not quite such good polyclonals. Company B here produces excellent mono and poly. And down here, company AD doesn't appear to produce anything good of any kind whatsoever. So he determined that the approximate ratio, he said that about 50% of antibodies were functional, 50% were non-functional. In a later assessment, he came up with 35% as the figure for antibodies that were functional. So 65% were non-functional. And then depending upon your application, Gavin McBeath in 2002, trying to do antibody arrays, found that only 5% of antibodies were able to function in antibody arrays. Now, this is slightly different. I think here what we're seeing is antibodies that actually aren't functional. Here is maybe more so antibodies that are non-functional for a particular application. And so depending upon the specific assay, only 5 to 49% of commercial antibodies appear to work. And so this is a fellow called John Wanamaker. He came up with this famous pithy aphorism: half the money I spent on advertising is wasted. The problem is I don't know which half. So if you knew in advance which antibodies weren't going to work, you could, of course, get 100% good antibodies. And here's another example. This antibody sold as recognizing CDK2, also recognizes this other target, CEP152. And you can see here that these two antibodies here, you see the recognition of this CEP52 in these two antibodies, but not in these ones here. This antibody has been used in over 200 published papers, and as a result of the reactivity of this antibody, theories about the interaction between these two proteins are being produced. So now I'm going to give you a little detour to how much of the research antibody business works. And probably my understanding of this is rather naive. I'm an academic, I'm not in the business. Maybe Roberto, who will speak later, can say a bit more. So there are over 300 vendors listed on antibodyresource.com, which is one of many websites that gives a list of tons of different targets and antibodies available against them. There are over 2 million antibodies listed on Site AB. Many antibody companies set up their development manufacturing sites overseas to take advantage of labor and other resource cost advantages. So, I mean, you can clearly make antibodies cheaper if you're elsewhere. Many antibody producers sell their antibodies to multiple vendors who add their own labels, and then in turn, those same vendors will resell to each other and add their own labels again. So you may think you're being smart by shopping around, but you actually may end up with the same antibody. The greatest cost in producing an antibody is actually characterization. So I've heard an estimate of about $200 for a Western blot and $60 for an immunohistochemistry slide. And this is why very often, for some of those cheaper antibodies, it's relatively minimal. And also why characterization information has been shown to be historical and does not necessarily apply to the lot that you purchase. So this is just my understanding of how it works. So you have all these different primary antibody manufacturers on the left. Let's just take one of them. They sell now to different antibody vendors who in turn sell to each other. And then you, as a user, buy antibodies from 10 different vendors. And in fact, all you've done is you may have bought the same antibody 10 times. So you may be skeptical about this. But if you look at this is from Antibodypedia, 380, and I'm only looking at monoclonal antibodies here, 380 monoclonal antibodies against hexakinase 1, an antigen I just picked at random from 27 providers on Antibodypedia. So you see all these providers. You may find that with the exception of this one here, which has been rotated through 90 degrees, you will notice that these patterns of reactivity look awfully similar. The other thing you'll notice is that they're all selling antibodies in microliters. The cost is pretty similar, but of course you have no idea how much antibody you're actually getting. And then here's 4 other manufacturers also selling in microliters, with the exception of this antibody down here, where you can buy it in micrograms, but unfortunately it's been discontinued. So what you see is that there are a lot of companies here selling obviously exactly the same antibody because the data is the same, and that's the only way you can really find that out. But there are fortunately other manufacturers out there as well which are selling alternative antibodies. So that illustrates the fact that there are vendors that do make their own antibodies, and if you know where you're going, you can also find and get those different sorts of antibodies. So how do you choose a good antibody? So as I've mentioned, there are many antibodies against popular targets. There's over 3,000 different targets that have over 100 different antibodies. 40 proteins have over 1,000 antibodies. And I don't know if you go to a supermarket and you're presented with choices of 1,000 different sorts of cheese or milk or something, it actually gets a bit daunting. You really only want one antibody that works. You're not interested in 999. You just want one. The record is held by this target here with over 3,000 antibodies. And as I said, it's impossible to test all, and many of these are duplicates. So citations is one possible way, and this is the commonest way. I'm going to say very little about this, but Fridtjof Lund Johansson, who's speaking later, will say more. The Human Protein Atlas has over 20,000 antibodies against 14,000 different targets, and they present the primary data, which allows you to make your own mind up as to the quality of the antibodies that are presented. And they do a number of different sorts of tests. So just to go quickly through the citation issue. So this is data from Fridtjof, and what you can see is here is Santa Cruz provides a very nice website where it tells you the number of citations against different antibodies. These are against CDK2. You can see there are two antibodies, one here cited once, one here cited 690 times. And this is a procedure that Fridtjof developed. And the important thing to notice, I don't want to go into the details, is that the pattern of reactivity of these two antibodies appears to be identical. This is molecular weight here, this is signal here. Here you have cytoplasm, nucleus, organelles, and membranes, and you can see that you've got increase in cytoplasmic signal at the right size for CDK2, you've got a little bit in the nucleus. But if you plot these on the same scale, you'll see that in fact this antibody here, cited only once, has signals about 10-fold greater than this antibody here, which is cited 690 times. What this reflects, I think, is the tyranny of the first antibody to publish, or a first mover advantage, and that's why some companies will try and get an antibody out as fast as possible, because once that antibody has been cited once, people like some reassurance and they will buy this well-cited antibody. How much money is wasted? It's a number of different estimates of the size of the market, ranging from about $1.6 to $2.2 billion. If we assume that published antibody functionality is, as I said, let's say 50%, and assume that everybody is buying good antibodies as frequently as they're buying bad antibodies, it means about just over $1 billion is wasted worldwide, almost $500 million in the US, on antibodies that don't work. And this does not take into account, this is just the cost of the reagents. It's not the cost of the secondary reagents. It's not the wasting of graduate student time or professor time or retractions in papers or people trying to follow up publications that then turn out not to have been based on a good antibody. And so how much does it cost to make a good antibody? And this is a difficult figure to get hold of, but one way you can look at the grant numbers in the NIH Reporter where people have promised to make antibodies, and you take the number of dollars provided, divide that by the number of supplied antibodies, which should take everything into account, and that gives you a figure of about $30,000 to $90,000 per antibody. And you may say, well, wait a minute, I can just go out and get an antibody. All these companies are selling antibodies for $5,000 to $15,000. And it turns out it's true, but they just do this little bit in the middle, which excludes the two most expensive steps, which is antigen production at this end and antibody characterization at this end. I would point out that, if one was to dedicate oneself to trying to do the whole genome, it would be about $1 billion, which is less than a third of the full cost of the Human Genome Project. And of course, in these tightened financial times, such a project would never really be financed, but it would be recuperated in 4 to 8 years of money wasted on bad commercial antibodies. So how do you solve this problem? And this is a slide that Andreas Pluckton prepared. And so your average customer would say, we just need better antibodies. The average manufacturer will say people will not buy anything but traditional antibodies. Henry Ford said if I had asked people what they wanted, they would have said faster horses. And Steve Jobs said it's not the consumer's job to know what they want, and people don't know what they want until you show it to them. So what's the conventional solution? Basically, antibodies just need to be better characterized, or scientists should purchase antibodies from the most reliable manufacturers. And both of those things are true. However, the problem with this is that even good antibodies are well characterized are not defined or archived forever. So polyclonals are practically undefinable. Monoclonals are unknown molecular entities, and I'll talk about this in a minute. 35% have more than one expressed VH or VL gene, and deep sequencing has revealed ongoing mutation of variable regions where you have substitutions and deletions in the CDRs changing specificity. Cell lines may die out, evolve, or need recloning. Some institutions, and Scripps is an example, have discarded preserved hybridomas upon the retirement of a professor, and essentially resulting in the complete loss of valuable published antibody specificities. If those antibodies were sequenced, then those specificities and those antibodies would not have been lost. There is lot-to-lot variation. It's impossible to know if two batches are the same. Data sheets are historical and often do not correspond to the supplied lots. And this reflects the fact that antibodies at the moment are sold on the basis of what they purportedly recognize and not what they actually are. Here's an example of published antibodies with multiple chains. So here we have heavy chain non-productive, heavy chain productive, light chain non-productive, light chain productive. And you can see that in some cases we have non-productive chains being produced. These obviously will not affect binding activity, but here and here and here we have productive additional chains either produced by the light chain or the heavy chain, which will provide additional specificities that will not be what you think they are. And in fact, there is some evidence, and in one case, this one here, where karyotypic analysis was done, it was found that the reason this antibody had, this hybridoma was expressing two different light chains was because the hybridoma was derived from fusion of two spleen cells to a single partner myeloma cell. So Natalie D'Souza, who's the editor of Nature Methods, summarized this quite nicely, saying that antibody companies, and of course not all of them, but many, antibody companies sell products they say are the same that are actually different, and products they say are different that are actually the same. So we published this article in Nature with, as I said, over 100 different co-signatories, and there was a response to this provided by Roberto and Len, both of whom will be speaking later today. And what they pointed out was absolutely right, was that it's not just enough. There's nothing special about recombinant antibodies. Antibodies obviously have to be validated. And this was a point that, unfortunately, in the multiple iterations of editing that went on when you submit a paper to Nature, that got lost. It was the message was in there in the early drafts, and then it just basically got hacked away by the Nature editors. So it's clear that we responded to this and said quite clearly that antibodies should be characterized and validated. The fact that they're recombinant makes no difference whatsoever. They just need to be treated in the same way as anything else. But we do believe that antibodies should be expressed recombinantly and identified by unique barcodes. They're publicly available sequence. There is no better barcode than a publicly available sequence. So why is this important? Reproducibility. So I can use the same exact binder that you use. I can repeat your experiments with the same reagents even decades later. Good binders will become immortal and never lost. The genes can always be resynthesized providing the antibodies sequences are available online, then they can always be reproduced. And as binder citations accumulate, you'd expect a complete understanding of the property of a specific binder. It may turn out that it may be very good for some particular properties and not others, and then that can be built into the history and the knowledge about that binder if it's all referred back to a sequence, as opposed to getting an antibody on the basis of its supposed recognition specificity, where no history, if you like, is maintained. Complete characterization is also required only once. Why do I say that? Well, if you have a recombinant antibody and you make it again, and providing there are no batch-to-batch variations, I think it's important just to confirm the binding activity after the characterization has been carried out, and you can assume that other properties will be similar. Now, there are also other aspects to recombinant binders. You can do other things with them. You can express them intracellularly, turn proteins or processes off. You should be able to do affordable affinity purifications of targets, crystallizations, do orthogonal detection with secondary reagents, do fusions, retarget viruses, and of course the whole field of chimeric antigen receptors would have been impossible without recombinant antibodies. So if they're so good, why aren't companies selling recombinants? Well, in fact, they are. There are 5 non-therapeutic antibodies that are publicly disclosing the fact that they're selling recombinants, but they don't reveal the sequences. ABD Serotech and Creative Diagnostic use in vitro methods to generate their antibodies. Biosite, Reaffinity, and Abfinity are producing recombinant antibodies generated from immunized animals. Between them, these companies have hundreds of antibodies, and this is to be compared to the 2 million traditional antibodies total that I mentioned earlier. Absolute antibodies provides recombinant antibodies by synthesizing genes and mainly going to expired patent sequences. Abcan has 5,000 rabbit monoclonals, originally derived from epitomics. Because the rabbit hybridomas actually express rather poorly, they found the only way to produce large amounts of these antibodies was to reclone them as recombinants. although I don't think that they're saying yet that they are recombinants. Some recombinant antibodies are recloned and then sold as monoclonals, and we, in fact, licensed an antibody to Millipore. It's an antibody that recognizes the tyrosine sulfate specificity, which was pretty impossible to produce by traditional immunization. The reason being that tyrosine sulfate is, as a post-translational modification, is found in the secretory pathway, and all antibodies recognizing it were held up in the secretory pathway and could not get out. So Millipore sold this antibody that we derived by phage display and just called it a monoclonal. If you look at the references and track back, eventually you'll discover it's actually a human antibody and not a mouse antibody. Most other recombinant antibody producers have in fact opted for therapeutics, the reason being that it's just much more profitable. So how many antibodies do you need per protein? So if all you're doing is a Western blot, then one will be enough. But if you think about a complicated protein like HER2, you could imagine there could be 20 or 30 different useful epitopes, including post-translational modification sites, different processed forms, different conformational forms, different splice sites or domains, different folded or unfolded for different specificities. So one antibody per target may not be enough if you want to do much more with your antibodies. So what are the unsolved technical problems in this field? So the expression of antigen, the production of antigen, is still really a big deal. It's difficult. It requires trial and error. It's expensive, and the production of the production of antigen that is well folded is an artisanal process. You need to go through, try different things until eventually you get something that's right. Different antigen forms are probably going to be required for the same target. So, for example, you want denatured antigen for Western blots, you want fixed antigen for immunohistochemistry. If you are looking for phosphorylation sites or other post-translational modifications, then you want to modify those. And finally, you may want different activation states. Now, some antibodies will work in all different applications and assays, and obviously that's the best sort of antibody that you can get hold of, but many antibodies are not that good and actually will vary in their properties according to the assay that you're using. What about the binder? So we know about generating antibodies by immunization. I've been working for a long time in the generation and Andreas as well, and many of the other people that signed the commentary, in the generation of high-quality recombinant reagents using in vitro methods. And we believe that they could become far, far cheaper. The scope for cost reduction by automation has really only just started. And I think that with greater automation, one could reduce the cost of producing antibodies really by orders of magnitude, I would argue. But this is just the generation of the binder. It doesn't, as I say, address either the production of the target, which is the preceding expensive step, or the next step, which I'm going to talk about. And customers in general using recombinant antibodies tend to expect a higher level of quality control in terms of affinity. This is just something we tend to do, is to measure the affinity of the antibodies we generate, as well as the sequence. What about the characterization? Along with the supply of antigen, this is the greatest cost in the supply of reliable binding reagents. And this is presently extremely labor-intensive and hardly automated. And I think ideally where we want to go with this is, and it may be impossible to get there, but ideally what we'd want is a surrogate for commonly used assays that can be carried out in high throughput. So what we think, these are our 2 bullet points, that antibodies should be well characterized and validated, and antibodies should be expressed recombinantly and identified by unique barcodes, which are their publicly available sequences. So just concentrating on this just very briefly about the characterization. So standard characterization methods include ELISA, Western blotting, immunohistochemistry or immunofluorescence, flow cytometry, protein or peptide arrays, and knockout cell lines. siRNA is being used. CRISPR-Cas9 has just started. There aren't any published papers on the use of CRISPR-Cas9, but there are some companies now that have started doing it. And I believe that Abcam has made a big deal about the fact that they are now selling CRISPR-Cas9 validated antibodies. That will tell you, that will determine and confirm the specificity of an antibody, but it doesn't necessarily indicate if the antibody has any off-target effects. Recently, a paper was published by Aled Edwards and others looking at immunoprecipitation and mass spectrometry, where he starts off with an antibody, he does an immunoprecipitation, uses mass spec to identify the proteins that are immunoprecipitated, uses the peptide identification to identify the recognized proteins, and then carries out antibody evaluation. He gave a talk on this in Vancouver in June, where he said that the cost of this for doing, I think it was 50 antibodies or something, was in the order of $3 million. So this is unlikely to become a generally useful approach. It may be, if you look here, he says the IP gold standard is the intended antigen places in the top 3 of the most abundant proteins, to which a cynic might ask, well, what about the other 2? So you wonder, is it binding something else that perhaps shouldn't be bound, or is it something being bound by the matrix? Anyway, it's unlikely, I think, given the cost of this approach, that it's going to be widely used. Fritjof has developed this method, he'll be talking about it a bit later, which is a multiplex flow cytometry method, where you can end up with 1,700 different beads by labeling them with different colors and then putting either different antibodies or different targets on those. I won't say any more about that. What about the sequencing side of things? So I think that's something that needs to be done, but there's some issues there, because nobody's going to actually produce the sequences or provide the sequences of the antibodies they use unless they're forced to do so. And so we think that funding agencies need to get involved here. They're reluctant, but they could establish and maintain a database of binding sequences, or it could otherwise be just farmed out to something like GenBank or Uniprot or something like that. If the funding agencies were to insist that all antibodies used in research were sequenced, recombinantly expressed, and their public sequences were publicly available, that would go a long way to ensuring that people were able to use exactly the same antibodies used in other studies. If this was done, PIs would have to be informed of the requirement, I would say, with a long lead time, say 5 to 10 years, so that there's no excuses for saying, oh, you've sprung this on us too quickly. And I think probably this should be done in specialized centers for binder selection, sequencing, and characterization. And then if the sequences were provided broadly to the general population, and that would include the companies, then one would be sure that the same sequences were being used. What about publishers? So likewise, we believe that antibodies used in published papers, if publishers were to insist that the sequences of those antibodies were to be published, and again, with a long lead time, then again, this would push people to use sequence recombinant antibodies. PEDS has said that they will be doing this within a couple of years. It's clear that beyond the sequence of the antibodies, you also need methods description that include binder concentrations in micrograms per ml, or ideally in nanomoles, and buffers as well. And the binder accession numbers should be provided in publications, ideally in a separate field, which will allow automatic archiving of all publications that use a particular antibody sequence. So going back to the response to our original commentary, Roberto writes here, even if the practical hurdles of funding and enforcing a sequencing publishing policy could be overcome, making unpatented antibody sequences public would allow them to be widely copied, produced, and sold. And I think this is a very valid concern. Companies put a lot of money into validating and characterizing their antibodies, and you can't expect them to put the sequences of those antibodies out. You can't expect them to put the sequences of those antibodies out to allow companies elsewhere that haven't put in any of the work to just copy them and make them again. So these are the concerns of the companies, that antibody sequences represent IP. If they publish them, others will use them. Research antibodies alone actually don't make enough money to patent. It's just not worth patenting. And so they will be unprotected. And I've looked into this, and there really are no cheap ways to protect antibody sequences. I thought maybe that copyright might be the way, but it just doesn't work. The only way to protect an antibody is by patenting, and that will just that's just too expensive. So some companies are saying that research antibodies don't make much money, and so it's not worth investing in the additional technology to make recombinants. If all you're doing is immunizing rabbits or mice, I can understand that's true. But many companies making antibodies these days actually have rather more sophisticated operations involving the production of recombinant targets. And so this should not really be an issue. There are some companies that claim they would have no problem publishing sequences, although whether they actually do so with the respect of absolute antibodies, which is still a very small company, and we'll have to wait and see. I think there's probably two ways to address this. One is the funding of public or public-private specialized centers that will carry out the selections and publish the sequences and characterize them so that the companies are not on the hook, if you like, for losing IP due to characterization they've done. An alternative is a disruptive technology that was able to generate, characterize, and produce antibodies so cheaply that the publication of sequences now no longer becomes a concern. And so oligonucleotides, if you like, is a similar, it could be likened to this, but this would require a significant reduction in all aspects of the chain that I've described. So antigen production would have to be reduced, binder selection would have to be reduced, and characterization of antibodies would have to become significantly cheaper. And I'd just like to end on the music industry as a victim of disruption. Okay, so here on the left we have millions of units, and here we have the millions of inflated adjusted dollars. So what we have here is we started off back in I think this is the 70s, I think this is 40 years here. So in the 70s we had LPs, and I remember singles and LPs. Never really was terribly fond of 8-tracks, but then onto cassettes, and I recorded many of my friends' LPs onto cassettes. And then at about 1980-something, CDs came in, and they took over the market. And then well, this must be about 10 years ago, downloads started, and you can see that downloads have increased as well. What you'll see is that pretty much with each new phase, there's been an increase in the number of units that have been consumed, let us say. But if you look at the millions of inflation-adjusted dollars, you'll see it reaches a peak at about 15 years ago with CDs, and then ever since then, it's been going down significantly. And what about streaming? And so I picked this up from this website down here, but there was nothing about streaming. This was done in 2000, this is data for 2013. And if you look at streaming, what I've done is I've taken this one and I've squashed it down. What you can see is this little thing here is the equivalent of that up there. And streaming, you can see, vastly dominates the whole picture. Okay, so here, in 2014, almost 80 billion songs were streamed, as opposed to consumed down here, I think is about 1 billion. But again, it's not helping the revenue, you see. The revenue is still not terribly good. So industries are subject to disruptive technologies. I do feel that the research antibody market is also ready for a disruption, but exactly how it comes, I'm not quite sure. Are they changing? Well, there's 3 organizations, FASEB, HUPO, and GBSI, that have established committees to look at the role of antibodies in research use. There's going to be a meeting, a workshop in California, in Asilomar, in fall of next year, hoping to address this. And we're all stakeholders, including funders, journals, antibody producers, research scientists, database providers, and so on will be invited. So in conclusion, I believe, notwithstanding the problems, and I think we need to find a solution, that we should apply the gene-based paradigm to antibodies. I think it's time for a new business model. One way this could be solved is with the presence of specialized centers. And ultimately, what I would hope, and many of the people on that commentary as co-signatories would hope, is that using an unsequenced antibody in the future will become as unacceptable as using an unsequenced plasmid, oligo, or gene. And with that, I'll stop. Thanks very much, and take questions.
The sequencing antibody is obviously a good idea, but who is going to sequence and how the sequence will be confirmed?
So you're absolutely, that's an issue. It's an issue that needs to be addressed. And that's why I think that ultimately I suspect that probably the best way to do this is with public funding or public-private partnerships, a bit like the Structural Genomics Consortium. Because why should companies sequence their antibodies in order to cut their own throats by releasing those sequences? I mean, it just doesn't make sense. I understand the business aspect of this. No, but they don't release the sequences, though. Yeah, right. That's what I'm saying. Jim, did you have?
Yes, I think maybe it's an oversimplification. You know, everybody buys these reagents because it saves time and they can put the money in their grants to buy them. And I don't think that will change if you have a real paradigm change and you can buy sequenced antibodies on a large scale and the prices aren't significantly different, you know, 50% more or something. And so I think people will still buy them because time is still, you know, they're competing. They don't want their graduate students to spend their time making antibodies.
That's right.
They'll find out how hard it is.
But if publishers were to stay and funding agencies were to say, look, from now on we want you to use sequenced antibodies, then I think the thing would change.
Yes, I think so. But I think the system would sort of re-equilibrate and
Right.
you know, people would still make money and people wouldn't go around spending all their time stealing the sequences.
Right.